Hello everyone, and welcome to today's webinar, Modeling of Cellular Interactions in Mixed Murine Organoids. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Andor. To learn more, please visit andor.oxinst.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Saskia Suijkerbuijk, Assistant Professor at Utrecht University. Saskia, you may now begin your presentation.
First of all, I would like to thank Andor for this opportunity to present our work. I recently started my lab at Utrecht University, and most of this work was done when we were still at the Medical Center Utrecht in Amsterdam, under the wings of Jacco van Rheenen. And what I would like to show today is how competitive cell interactions can influence oncogenic growth in mixed intestinal organoids. Within tissues, cells continuously compare their fitness with that of their neighboring cells. Weaker or looser cells are identified by stronger winner cells and eliminated from these tissues, often through apoptosis. And this is a strong mechanism that controls overall tissue and organismal fitness, which was first identified in developing fly tissues in the 70s. But it can act as a regulator of quality control throughout organismal life. And this starts in early embryo and continues to impact physiology up to late adulthood. But cell competition has also been shown to protect against disease. For example, by elimination of early malignant cells from pancreatic or intestinal epithelia, or through cell selection in the thymus, which can protect against leukemia. However, on the other side of the spectrum, we and others have shown that cancer cells can use cell competition for their own benefit. And this is exactly what my lab is currently focused on. And this is all driven by differences in cellular fitness. And what this means, I would like to illustrate by two pieces of work that I contributed to as a postdoc in the lab of Eugenia Piddini in the Gurdon Institute in Cambridge, where we studied cell competition in the intestine of the fruit fly. And what we found is that cells that have a ribosome impairment, also known as minute cells, are eliminated by surrounding wild-type enterocytes. However, the same wild-type enterocytes themselves are eliminated when they are facing APC mutant cells. And this is super interesting because genetically these wild-type cells are identical, but their survival is fully dependent on the fitness of their neighbors. In order to study such competitive behavior of cell populations, we set out to develop a model system based on murine intestinal organoids. And what we do is first derive organoid lines with a different cellular fitness, for example of a wild type and of a cancer origin. And next we label these cells differentially with fluorescent markers. These cultures are then broken into either pieces or single cells, mixed and formed into 3D mixed organoids or enteral monolayers. And these populations can then be followed over time on both a population or a cellular level. And for now, I will first focus on the mixed enteroid monolayers. We decided to use these to evaluate a role for cell competition in colorectal cancer. And therefore, we derived small intestine organoids of a wild-type nature that are labeled in magenta throughout this presentation. And we derived organoids of a cancer origin that lack APC, are p53 mutant, and express oncogenic KRAS. And these will be labeled in green throughout. And an example of such a monolayer is shown here on the right. When we analyzed the behavior of these different cell populations in these cultures, we observed something interesting. Because where pure populations of wild-type cells happily expand and mature, instead in mixed populations, this wild-type population is gradually lost over time. When we have a closer look at these monolayers, we observe that these wild-type cells reduce both in size, and an example is outlined here in cyan on the right, and in number, and a cell that is disappearing is shown in magenta on the right. And therefore, this indicates that these cells are eliminated from these monolayers. And this prompted us to have a closer look at and evaluate markers of cell death, such as cleaved caspase 3. What we found is that where pure populations of either wild type or cancer cells showed very little activation of this marker of cell death, instead there's a marked increase in the amount of cleaved caspase positive wild type cells in mixed conditions, indicating that these cells are eliminated by cancer cells in an apoptosis dependent manner. Of course, we were very interested to see what the influence of cell competition is on colorectal cancer in near-native conditions, and therefore we next turned to these 3D mixed cultures. And for this, we used the same wild-type population that we previously used in the entered monolayers that again are labeled in magenta, and the same cancer population that are marked in green. But instead, we generated these 3D mixed cultures. And an example of this is shown here on the right. First, I would like you to focus on the wild-type population in these cultures. Because what we observed is that where pure populations happily grow, they mature, they expand, they form these crypt-like structures exactly what you would expect from a wild-type intestinal population. Instead, the mixed wild-type population is lost from these cultures over time, which is shown in the decrease here in the graph on the right. In fact, when we quantify the contribution of the wild-type population to these organoids over time, we observed that there's a lower amount of wild-type cells at the end of the experiment than there is at the beginning of the experiment. And this showed us that the wild-type population is actively eliminated from these organoids, and it's not just a mere passive difference in proliferation rate. In order to characterize what happens to these cells, we had a closer look at the fate of these wild types. And we found that cells were extruded from the epithelia, and that there's an accumulation of wild-type cell debris in the lumen, indicating that these cells are eliminated through cell death. And therefore we had a closer look at markers of apoptosis. And what we found is that in the mixed populations there's an increase in the amount of cleaved caspase positive cells, showing that they are indeed eliminated through apoptosis. And interestingly, we could see activation of this marker both at the interface with cancer cells, but also further away in the inner cell mass. And even though elimination of wild-type cells can be induced at a distance from these cancer cells, multiple lines of evidence suggest that cell competition is actually short-ranged. Because what we found is that, first of all, the wild-type population and the cancer population are part of the same epithelium, and these competing cell populations directly interact and closely intermingle. Second, we could observe occasional organoids of a pure wild-type origin nearby mixed organoids, suggesting that elimination of wild-type cells depends on intra-organoid interactions with cancer cells. And lastly, the growth and the behavior of pure wild-type conditions that are plated in the same well with pure cancer or mixed organoids were undistinguishable of solely pure wild-type cultures, indicating that the presence of both cell populations within the same organoid is required for outcompetition of wild-type cells. Importantly, when we next treated these mixed cultures with a pan-caspase inhibitor, this could protect the wild-type population, and it increases the contribution of the wild-type population to mixed organoids. And therefore, this altogether told us that intestinal cells are actively eliminated by cancer cells through apoptosis. So far, I have shown you that the presence of cancer cells has a negative influence on wild-type intestinal cells. But is there an effect of these competitive interactions on the cancer population as well? And therefore, I would like to shift your attention to the other population in these cultures. And what we observed is that there's a higher expansion rate of cancer cells in mixed conditions compared to the behavior or the expansion rate of cancer cells in pure conditions, suggesting that there might be a proliferative advantage for cancer cells by the elimination of wild types. And to further dive into this, we looked at different markers of cell cycle progression, and we found that cancer cells have a higher rate of cycling cells in mixed conditions. And we observed that cells are more likely to progress from S phase to mitosis in a period of 4 hours, and that there are higher rates of mitotic cells. And altogether, this shows us that cancer cells use competitive cell interactions to boost their growth. What I've shown you so far is that competitive cell interactions shape the behavior of both wild-type and cancer cells when they're in mixed conditions. And to get a better understanding of what drives these interactions, we decided to sort wild-type cells from pure and mixed conditions, and next subject these cells to bulk mRNA sequencing. What we found is that overall cell competition induces expression of genes that are involved in cell death, while genes that are involved in cell cycle and metabolism are repressed. And this was very reassuring, because of course this is exactly what you would expect from a population that is being outcompeted. However, when we started to have a look at the individual genes that were differentially expressed in these mixed populations, we really got excited, because this is what caught our attention is the increased expression of two particular gene families, that of Ly6 and annexin. And this was particularly interesting to us, because in the past years, multiple labs have shown that these genes are part of a signature that is induced in intestinal tissues upon different damaging insults. And what they showed is that, for example, colitis or infection with parasitic helminths can cause reversion of cells to a fetal-like state. And this helps the tissue to cope with this stress. And indeed, when we compared our dataset to a previously published dataset, we found a clear enrichment, indicating that cell competition might cause a similar response and induce this fetal-like state or damaging state in intestinal wild-type cells. To further investigate this response, we went back to our mixed cultures, and we found that a main marker of this response, Ly6, which is also known as SCA1, is highly expressed in mixed wild-type cells, shown here on the picture in the middle, but also in the quantification on the right. While pure wild-type cells are completely devoid of this expression of this marker. And this indicates that indeed wild-type cells revert to a primitive fetal-like state when they are in close proximity of cancer cells. Another characteristic of this response is the loss of cell types that are normally present in adult intestinal tissues, such as stem cells. And interestingly, in our mixed cultures, we observed that the wild-type population undergoes a morphological change. It loses these typical crypt-like structures that are normally present in intestinal organoids. And this also coincides with a reduction in the number of cells that are positive for the stem cell marker LGR5, confirming that during competition, mature cell types are lost in the wild-type population, and these cells revert to a primitive fetal-like state. Well, because this happens and we see this reversion, we speculated that possibly cell state transition might contribute to competition, and therefore we had a look and further investigated how we could possibly manipulate this. And one of the methods that we tried is to a previously reported treatment of chiron and valproic acid. And what has been shown in the past is that this could enrich for stem cells in intestinal cultures. And indeed, also in our hands, we saw a massive increase in the amount of LGR5 positive cells, shown in the pictures here. So this is with treatment in the middle, but also in the quantification on the right. Of course, we next wondered whether this had a functional effect on competition and the phenotypes that we observed before. And therefore, we had a look at activation of SCA1, and we found that enrichment of the stem cell population could prevent the induction of SCA1 and in mixed wild-type cells, indicating that indeed this combined treatment of chiron and valproic acid can prevent this transition to a fetal-like state. Importantly, when we next evaluated the impact of prevention of this cell state transition to the outcome of competition, we found that this is sufficient to protect the wild-type populations and prevent their elimination, as confirmed by the quantification here. Importantly, this prevention of competition did not only influence the wild-type population, but it could also take away the competitive advantage of cancer cells and limit their overproliferation rate. And together this told us that during competition, wild-type cells revert to a primitive cell state, which can be used by cancer cells to promote their proliferative capacity. Next, to get insight into the mechanisms that might drive this, we went back to our mRNA sequencing data. And what we found is that AP1 sites are enriched in genes that are differentially expressed in mixed wild-type cells. And these AP1 sites are bound by a heterodimeric transcription factor complex containing, for example, CJON. And therefore, we next wondered whether there's a differential behavior of this particular transcription factor in competing cells. And we use this phosphospecific antibody that recognizes the activated form of the protein and can be recognized by nuclear localization. And what we observed is that cancer cells show a heterogeneous high expression of high activation of CJON, irrespective of competition. We can observe this in the pure conditions, but also in the mixed conditions. However, what we found is that there's little activation of CJON in pure wild-type cells. But there's an increased activation of this nuclear transcription factor in mixed wild-type populations, also reflected here in the quantification on the left. Interestingly, when we had a closer look at these phospho-JON positive cells, we saw that the distance to cancer cells is lower, and that there's an enrichment of activated cells, wild-type cells, that are in close proximity of cancer cells, together indicating that short-range signals from cancer cells can be involved in the activation of CJON. And this activation was particularly interesting to us, because CJON is a downstream target of J&K kinase, which is activated as a response to a wide range of cellular stressors, but is also known as a regulator of many different types of cell competition. And therefore, we next wondered whether J&K signaling is involved in cell competition in this system. And what we did first is treat our cultures with a small molecule that specifically inhibits the J&K kinase. And this treatment was sufficient to prevent the reversion of wild-type cells to a fetal-like state upon competition, giving us the first indication that J&K might be involved in outcompetition of wild-type cells. Next, what we found is that J&K inhibition could prevent the elimination of wild-type cells, therefore confirming a role for J&K signaling in cell competition in intestinal organoids. However, treatment with a small molecule inhibitor targets both cancer cells and wild-type cells. And indeed, when we have a look at phospho-JON in these populations, we see that treatment with this inhibitor completely abolishes the staining with this phosphospecific antibody in wild-type cells, also shown here, but also in the cancer population, shown here. And therefore, with an experiment like this, we are unable to untangle in which of these cell populations J&K activation is involved. So therefore, we designed a method that we can use to specifically inhibit J&K signaling in individual cell populations. And what we decided to do is use inducible expression of a phosphatase that can inhibit the J&K kinase, known as MKP5 or DOSP10. And using a doxycycline inducible system, we can thereby target J&K signaling in either cancer cells or in wild-type cells. And first, we expressed MKP5 only in the cancer population. And what we saw is that treating these cultures with doxycycline did not affect competition at all. The amount of wild-type cells that are present in these organoids is unchanged and the ratio is not affected, indicating that J&K signaling is not needed in the cancer population. However, when we next inhibited J&K activation solely in the wild-type population, we saw a major striking effect because this prevented the elimination of the wild-type population, showing us that cell competition activates J&K in wild-type cells and that this is required for their elimination. In summary, what I've shown today is that wild-type intestine cells are actively eliminated by cancer cells, and this results in induction of a fetal-like state which is driven by J&K signaling in the wild-type population. Importantly, inhibition of competition through preventing the cell state transition or through inhibiting J&K signaling can protect the wild-type population, but it also limits the oncogenic growth of cancer cells, and which of course opens up a very interesting window for treatment options, because instead of targeting tumor cells directly with the possible side effects that come with this, instead we can envision that inhibiting competition can simultaneously protect the surrounding healthy tissue while limiting tumor growth. And with this, I would like to close by thanking a lot of people. First of all, I recently started my lab at Utrecht University, but most of this work was done under the wings of Jacco van Rheenen at the Netherlands Cancer Institute, with the help of a lot of collaborators that is shown here on the right. And it was financially supported by the Dutch Cancer Society. And in particular, I would like to thank Anna Grotenberg-Garcia, who did a large part of this work and kindly joined me in Utrecht when I moved my lab there. Thank you.
Thank you, Saskia, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right, let's get started. Our first question here asks, is there a possibility to adjust the ratio of the different cell populations in mixed organoids?
Hi, Kaylee. Thank you for moderating the session and for this question. This is a very interesting part of the procedure that we use, and practically it is a bit difficult because it really depends on the behavior of the individual cell populations themselves. In case they are relatively equal in fitness, the ratio is very easy to adjust because then you can play with single cell suspensions. In the case of the small intestine, we really needed to work with pieces of organoids to generate these mixed organoids, which makes it a lot more difficult to work with different actual ratios. Of course, by using live imaging, one can visualize organoids from the start with different ratios and follow those over time, which would solve this question.
Great, thank you. All right, our next question here asks, which kind of microscope do you use to image mixed organoids in 3D?
Yes, we actually use two different types of microscopes, depending on the question that we would like to answer. So for live imaging, which is a large part of our work, we love to use spinning discs. And our favorite one that we did most of the imaging on is the dragonfly from Andor. So this really allows rapid imaging over time in 3D. For the fixed images, we normally use scanning confocals. Depending on the question to be asked.
Great, thank you. All right, our next question here says, related to the previous question, how do you visualize the data after acquisition?
Yeah, this is, of course, very important as well. Microscopy is not just dependent on the microscope that you use, but also a lot of it is data analysis afterwards. So especially when we're talking about time-lapse imaging, this is massive amounts of data. And we find that the best way to process this is using software that can do 3D reconstructions of these different Z stacks that you acquire over time. And most of this work was done using the IMARES software from Bitplane, which is now Oxford Scientific, I think.
Great. We have another question here that asks, is the observed phenotype specific to colorectal cancer with this specific combination of mutations?
Yeah, so this is something we, of course, we're wondering as well. You want to make sure that this is not just something that you see with one particular type of cells, but in fact we've used multiple different combinations of mutations, and they all show similar phenotypes, meaning that we can see that these different types of cancer cells can eliminate wild-type cells in an apoptosis-dependent manner, and they all induce a fetal-like state.
Great, thank you. It looks like we have another question here that asked, can engraftment of cancer cells onto wild-type cell layers be prevented by increasing fitness of wild-type cells?
Yeah, this is super interesting, of course, in particular when you're thinking about treatment options. And we think you can, based on the data that we have using apoptosis inhibitors, for example, or inhibiting of J and K signaling, because this basically prevents the outcompetition of these wild-type cells, but also makes them stronger. And this also prevents the overgrowth of cancer cells in this situation. So there is a possibility that this definitely works, yeah.
All right, well, we'll go ahead and wrap up with that final question there. Thank you again, Saskia, for your time today and your important research. We'd also like to thank LabRoots and our sponsor, Andor, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Bye-bye.